Section 15 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by M.L. Cohen, Anatomy of the Human Body Part 4 by Henry Gray. The Forebrain, Part 3. Temporal lobe, lobus temporalis. The temporal lobe presents superior, lateral, and inferior surfaces. The superior surface forms the lower limit of the lateral fissure and overlaps the insula. On opening out the lateral fissure, 3 or 4 gyri will be seen springing from the depths of the hinder end of the fissure and running obliquely forward and outward on the posterior part of the upper surface of the superior temporal gyrus. These are named the transverse temporal gyri. Heschel. The lateral surface is bounded above by the posterior ramus of the lateral fissure and by the imaginary line continued backward from it. Below, it is limited by the inferior lateral border of the hemisphere. It is divided into superior, middle, and inferior gyri by the superior and middle temporal sulci. The superior temporal sulcus runs from before backwards across the temporal lobe and some little distance below, but parallel with the posterior ramus of the lateral fissure, and hence it is often termed the parallel sulcus. The middle temporal sulcus takes the same direction as the superior, but is situated at a lower level and is usually subdivided into 2 or more parts. The superior temporal gyrus lies between the posterior ramus of the lateral fissure and the superior temporal sulcus and is continuous behind with the supramarginal and angular gyri. The middle temporal gyrus is placed between the superior and middle temporal sulci and is joined posteriorly with the angular gyrus. The inferior temporal gyrus is placed below the middle temporal sulcus and is connected behind with the inferior occipital gyrus. It also extends around the inferior lateral border onto the inferior surface of the temporal lobe, where it is limited by the inferior sulcus. The inferior surface is concave and is continuous posteriorly with the tentorial surface of the occipital lobe. It is traversed by the inferior temporal sulcus, which extends from near the occipital pole behind to within a short distance of the temporal pole in front, but is frequently subdivided by bridging gyri. Lateral to this fissure is the narrow tentorial part of the inferior temporal gyrus, and medial to it the fusiform gyrus, which extends from the occipital to the temporal pole. This gyrus is limited medially by the collateral fissure, which separates it from the lingual gyrus behind and from the hippocampal gyrus in front. The insula, island of Ryle central lobe, lies deeply in the lateral or sivulant fissure, and can only be seen when the lips of that fissure are widely separated, since it is overlapped and hidden by the gyri which bound the fissure. These gyri are termed the opercula of the insula. They are separated from each other by the 3 rami of the lateral fissure, and are named the orbital frontal, frontotemporal, and temporal opercula. The orbital operculum lies below the anterior horizontal ramus of the fissure, the frontal between this and the anterior ascending ramus, the parietal between the anterior ascending ramus and the upturned end of the posterior ramus, and the temporal below the posterior ramus. The frontal operculum is of small size in those cases where the anterior horizontal and ascending rami of the lateral fissures arise from a common stem. The insula is surrounded by a deep circular sulcus, which separates it from the frontal, parietal, and temporal lobes. When the opercula have been removed, the insula is seen as a triangular eminence, the apex of which is directed toward the anterior perforated substance. It is divided into a larger anterior and a smaller posterior part by a deep sulcus, which runs backward and upward from the apex of the insula. The anterior part is subdivided by a shallow sulci into 3 or 4 short gyri, while the posterior part is formed by 1 long gyrus, which is often bifurcated at its upper end. The cortical gray substance of the insula is continuous with that of the different opercula, while its deep surface corresponds with the lentiform nucleus of the corpus striatum. Limbic lobe. The term limbic lobe was introduced by Broca, and under it he included the cingulate and hippocampal gyri, which together arch around the corpus callosum in the hippocampal fissure. These he separated on the morphological ground that they are well developed in animals possessing a keen sense of smell, osmotic animals, such as the dog and the fox. They were thus regarded as part of the Rhinencephalon, but it is now recognized that they belong to the Neopallium. The cingulate gyrus is therefore sometimes described as part of the frontal lobe, and the hippocampal as part of the temporal lobe. The cingulate gyrus, gyrus cinguli, callosal convolution, is an arch-shaped convolution lying in close relation to the superior surface of the corpus callosum, from which it is separated by a slit-like fissure, the callosal fissure. It commences below the rostrum of the corpus callosum, curves around in front of the genu, extends along the upper surface of the body, and finally turns downward behind the splenium, where it is connected by a narrow isthmus with the hippocampal gyrus. It is separated from the medial part of the superior frontal gyrus by the cingulate sulcus, and from the precuneus by the subparietal sulcus. The hippocampal gyrus, gyrus hippocampi, is bounded above by the hippocampal fissure and below by the anterior part of the collateral fissure. Behind, it is continuous superiorly through the isthmus with the cingulate gyri and inferiorly with the lingual gyrus. Running in the substance of the cingulate and hippocampal gyri and connecting them together is a tract of arched fibers named the cingulum. The anterior extremity of the hippocampal gyrus is recurved in the form of a hook, uncus, which is separated from the apex of the temporal lobe by a slight fissure, the incisura temporalis. Although superficially continuous with the hippocampal gyrus, the uncus forms morphologically a part of the Rhinencephalon. The hippocampal fissure, fissura hippocampi, dentate fissure, begins immediately behind the splenium of the corpus callosum and runs forward between the hippocampal and dentate gyri to end in the uncus. It is a complete fissure and gives rise to the prominence of the hippocampus in the inferior cornu of the lateral ventricle. Rhinencephalon. The rhinencephalon comprises the olfactory lobe, the uncus, the subcallosal and supracallosal gyri, the fascia dentata hippocampus, the septum pellucidum, the fornix, and the hippocampus. 1. The olfactory lobe, lobus olfactorius, is situated under the inferior or orbital surface of the frontal lobe. In many vertebrates, it constitutes a well-marked portion of the hemisphere and contains an extension of the lateral ventricle. But in man, and some other mammals, it is rudimentary. It consists of the olfactory bulb and tract, the olfactory trigone, the para-olfactory area of Broca, and the anterior perforated substance. A. The olfactory bulb, bulbus olfactorius, is an oval reddish-gray mass which rests on the cribriform plate of the ethmoid and forms the anterior expanded extremity of the olfactory tract. Its undersurface receives the olfactory nerves which pass upward through the cribriform plate from the olfactory region of the nasal cavity. Its minute structure is described later. B. The olfactory tract, tractus olfactorius, is a narrow white band triangular on coronal section, the apex being directed upward. It lies in the olfactory sulcus on the inferior surface of the frontal lobe and divides posteriorly into two striae, a medial and a lateral. The lateral stria is directed across the lateral part of the anterior perforated substance and then bends abruptly medialward towards the uncus of the hippocampal gyrus. The medial stria turns medialward behind the para-olfactory area and ends in the subcallosal gyrus. In some cases, a small intermediate stria is seen running backward to the anterior perforated substance. C. The olfactory trigone, Trigonum olfactorium, is a small triangular area in front of the anterior perforated substance. Its apex, directed forward, occupies the posterior part of the olfactory sulcus and is brought into view by throwing back the olfactory tract. D. The para-olfactory area of Broca, Area para-olfactoria, is a small triangular field on the medial surface of the hemisphere in front of the subcallosal gyrus, from which it is separated by the posterior para-olfactory sulcus. It is continuous below with the olfactory trigone and above and in front with the cingulate gyrus. It is limited anteriorly by the anterior para-olfactory sulcus. E. The anterior perforated substance, substantia perforata anterior, is an irregularly quadrilateral area in front of the optic tract and behind the olfactory trigone, from which it is separated by the fissura primea. Medially and in front, it is continuous with the subcallosal gyrus. Laterally, it is bounded by the lateral stria of the olfactory tract and is continued into the uncus. Its gray substance is confluent above with that of the corpus striatum and is perforated anteriorly by numerous small blood vessels. 2. The uncus has already been described as the recurved, hook-like portion of the hippocampal gyrus. 3. The subcallosal, supracallosal, and dentate gyri form a rudimentary arch-shaped lamina of gray substance extending over the corpus callosum and above the hippocampal gyrus from the anterior perforated substance to the uncus. A. The subcallosal gyrus, gyrus subcallosus, pedicome of the corpus callosum, is a narrow lamina on the medial surface of the hemisphere in front of the lamina terminalis, behind the paraolfactory area, and below the rostrum of the corpus callosum. It is continuous around the genu of the corpus callosum with the supracallosal gyrus. B. The supracallosal gyrus, indusium griseum, gyrus epicallosus, consists of a thin layer of gray substance in contact with the upper surface of the corpus callosum and continuously laterally with the gray substance of the cingulate gyrus. It contains two longitudinally directed strands of fibers termed respectively the medial and lateral longitudinal striae. The supracallosal gyrus is prolonged around the splenium of the corpus callosum as a delicate lamina, the fasciola cinerea, which is continuous below with the fascia dentata hippocampi. C. The fascia dentata hippocampi, gyrus dentatus, is a narrow band extending downward and forward above the hippocampal gyrus, but separated from it by the hippocampal fissure. Its free margin is notched and overlapped by the fimbria, the fimbrio-dentate fissure, intervening. Anteriorly, it is continued into the notch of the uncus, where it forms a sharp bend and is then prolonged as a delicate band, the band of Giacomini, over the uncus on the lateral surface of which it is lost. The remaining parts of the Rhinencephalon, that is the septum pellucidum, fornix, and hippocampus, will be described in connection with the lateral ventricle. Interior of the cerebral hemispheres. If the upper part of either hemisphere be removed at a level about 1.25 centimeters above the corpus callosum, the central white substance will be exposed as an oval-shaped area, the centrum ovale minus, surrounded by a narrow convoluted margin of gray substance and studded with numerous minute red dots, puncta vasculosa, produced by the escape of blood from divided blood vessels. If the remaining portion of the hemisphere will be slightly drawn apart, a broad band of white substance, the corpus callosum will be observed connecting them at the bottom of the longitudinal fissure. The margins of the hemispheres which overlap the corpus callosum are called the labia cerebri. Each labrium is part of the cingulate gyrus already described, and the slit-like interval between it and the upper surface of the corpus callosum is termed the callosal fissure. If the hemispheres be sliced off to a level with the upper surface of the corpus callosum, the white substance of that structure will be seen connecting the two hemispheres. The large expanse of medullary matter now exposed, surrounded by a convoluted margin of gray substance, is called the centrum ovale majus. The corpus callosum is the great transverse commissure which unites the cerebral hemispheres and roofs the lateral ventricles. A good conception of position and size is obtained by examining a medial sagittal section of the brain. When it is seen to form an arch-straight structure about 10 centimeters long, its anterior end is about 4 centimeters from the frontal pole, and its posterior end about 6 centimeters from the occipital pole of the hemisphere. The anterior end is named a genu, and is bent downward and backward in front of the septum pellucidum. Diminishing rapidly in thickness, it is prolonged backwards under the name of the rostrum, which is connected below with the lamina terminalis. The anterior cerebral arteries are in contact with the undersurface of the rostrum. They then arch over the front of the genu and are carried backwards above the body of the corpus callosum. The posterior end is termed the splenium and constitutes the thickest part of the corpus callosum. It overlays the telechoroidea of the third ventricle and the midbrain and ends in a thick convex free border. A sagittal section of the splenium shows that the posterior end of the corpus callosum is actually bent forward, the upper and lower parts being applied to each other. The superior surface is convex from before backward and is about 2.5 centimeters wide. Its medial part forms the bottom of the longitudinal fissure and is in contact posteriorly with the lower border of the falx cerebri. Laterally, it is overlapped by the cingulate gyrus, but is separated from it by the slit-like callosal fissure. It is traversed by numerous transverse ridges and furrows and is covered by a thin layer of gray matter, the supracallosal gyrus, which exhibits on either side of the middle line the medial and lateral longitudinal striae already described. The inferior surface is concave and forms on either side of the middle line of the roof of the lateral ventricle. Medially, this surface is attached in front to the septum pellucidum. Behind this, it is fused with the upper surface of the body of the fornix, while the splenium is in contact with the telechoroidea. On either side, the The fibers of the corpus callosum radiate in a white substance and pass to the various parts of the cerebral cortex. Those curving forward from the genu to the frontal lobe constitute the forceps anterior, and those curving backward into the occipital lobe, the forceps posterior. Between these two parts is the main body of the fibers which constitute the tapetum and extends laterally on either side into the temporal lobe and cover in the central part of the lateral ventricle. The lateral ventricles. Ventriculus lateralis. The two lateral ventricles are irregular cavities situated in the lower medial parts of the cerebral hemisphere, one on either side of the middle line. They are separated from each other by a median vertical partition, the septum pellucidum, but communicate with the third ventricle and indirectly with each other through the interventricular foramen. They are lined by a thin diaphanous membrane, the ependyma, covered by ciliated epithelium, and contain cerebrospinal fluid, which, even in health, may be secreted in considerable amount. Each lateral ventricle consists of a central part or body and three prolongations from it termed cornua. The central part, pars centralis ventricula lateralis, cella, of the lateral ventricle extends from the interventricular foramen to the splenium of the corpus callosum. It is an irregularly curved cavity, triangular on transverse section, with a roof, a floor, and a medial wall. The roof is formed by the undersurface of the corpus callosum, the floor by the following parts enumerated in order of their position from before backward: the caudate nucleus of the corpus striatum, the stria terminalis and the terminal vein, the lateral portion of the upper surface of the thalamus, the choroid plexus, and the lateral part of the fornix. The medial wall is the posterior part of the septum pellucidum, which separates it from the opposite ventricle. The anterior cornu, cornu anterius, anterior horn, precornu, passes forward and lateralward with a slight inclination downward from the interventricular foramen into the frontal lobe, curving around the anterior end of the caudate nucleus. Its floor is formed by the upper surface of the reflected portion of the corpus callosum, the rostrum. It is bounded medially by the anterior portion of the septum pellucidum and laterally by the head of the caudate nucleus. Its apex reaches the posterior surface of the genu of the corpus callosum. The posterior cornu, cornu posterius post cornu, passes into the occipital lobe, its direction being backward and lateralward and then medialward. Its roof is formed by the fibers of the corpus callosum passing to the temporal and occipital lobes. On its medial wall is a longitudinal eminence, the calcar avris, hippocampus minor, which is an involution of the ventricular wall produced by the calcarine fissure. Above this, the forceps posterior of the corpus callosum sweeping around to enter the occipital lobe causes another projection, termed the bulb of the posterior cornu. The calcar avus and bulb of the posterior cornu are extremely variable in their degree of development. In some cases they are ill-defined, in others prominent. The inferior cornu, cornu inferior, descending horn, middle horn, medial cornu, is the largest of the three, transverses the temporal lobe of the brain, forming in its course a curve around and posterior end of the thalamus. It passes at first backward, lateralward, and downward, and then curves forward to within 2.5 centimeters of the apex of the temporal lobe, its direction being fairly well indicated on the surface of the brain by that of the superior temporal sulcus. Its roof is formed chiefly by the inferior surface of the tapetum of the corpus callosum, but the tail of the caudate nucleus and the stria terminalis also extend forward in the roof of the inferior cornu to its extremity, where they end in a massive gray substance, the nucleus amygdalae. Its floor presents the following parts: the hippocampus, the fimbria hippocampi, the collateral eminence, and the choroid plexus. When the choroid plexus is removed, a cleft-like opening is left along the medial wall of the inferior cornu. This cleft constitutes the lower part of the choroidal fissure. End of section 15. Section 16 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information to volunteer, please visit librivox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Forebrain Part 4. The hippocampus, hippocampus major, is a curved eminence about 5 centimeters long, which extends throughout the entire length of the floor of the inferior cornu. Its lower end is enlarged and presents 2 or 3 rounded elevations or digitations, which give it a paw-like appearance, and hence it is named a pes hippocampi. If a transverse section be made through the hippocampus, it will be seen that this eminence is produced by the folding of the wall of the hemisphere to form the hippocampal fissure. The main mass of the hippocampus consists of gray substance, but on its ventricular surface is a thin white layer, the alveus, which is continuous with the fimbria hippocampi. The collateral eminence, eminentia collateralis, is an elongated swelling lying lateral and parallel with the hippocampus. It corresponds with the middle part of the collateral fissure, and its size depends on the depth and direction of this fissure. It is continuous behind with a flattened triangular area, the trigonum collaterale, situated between the posterior and inferior cornua. The fimbria hippocampi is a continuation of the crus of the fornix and will be discussed with that body. A description of the choroid plexus will be found later. The corpus striatum has received its name from the striped appearance which a section of its anterior part presents, in consequence of diverging white fibers being mixed with the gray substance which forms its chief mass. A part of the corpus striatum is embedded in the white substance of the hemisphere and is therefore external to the ventricle. It is termed the extraventricular portion or the lentiform nucleus. The remainder, however, projects into the ventricle and is named the interventricular portion or the caudate nucleus. The caudate nucleus, nucleus caudatus, caudatum, is a pear-shaped, highly arched gray mass. Its broad extremity or head is directed forward into the anterior cornua of the lateral ventricle and is continuous with the anterior perforated substance and with the anterior end of the lentiform nucleus. Its narrow end, or tail, is directed backwards on the lateral side of the thalamus, from which it is separated by the stria terminalis and the terminal vein. It is then continued downward into the roof of the inferior cornua and ends in the nucleus amygdalae at the apex of the temporal lobe. It is covered by the lining of the ventricle and crossed by some veins of considerable size. It is separated from the lentiform nucleus in its greater part of its extent by a thick lamina of white substance called the internal capsule, but the two portions of the corpus striatum are united in front. The lentiform nucleus. Nucleus lentiformis, lenticular nucleus, lenticula, is lateral to the caudate nucleus and thalamus and is seen only in sections of the hemisphere. When divided horizontally, it exhibits to some extent the appearance of a biconvex lens, while a coronal section of its central part presents a somewhat triangular outline. It is shorter than the caudate nucleus and does not extend as far forward. It is bounded laterally by a lamina of white substance called the external capsule, and lateral to this is a thin layer of gray substance termed the claustrum. Its anterior end is continuous with the lower part of the head of the caudate nucleus and with the anterior perforated substance. In a coronal section through the middle of the lentiform nucleus, two medullary laminae are seen dividing it into three parts. The lateral and largest part is of a reddish color and is known as the putamen, while the medial and intermediate area of a yellowish tint and together constitute the globus pallidus. All three are marked by fine radiating white fibers, which are most distinct in the putamen. The gray substance of the corpus striatum is traversed by nerve fibers, some of which originate in it. The cells are multipolar, both large and small. Those of the lentiform nucleus contain yellow pigment. The caudate and lentiform nuclei are not only directly continuous with each other anteriorly, but are connected to each other by numerous fibers. The corpus striatum is also connected, 1. to the cerebral cortex by what are termed corticostriate fibers, 2. to the thalamus by fibers which pass through the internal capsule, and by a strand named ansa lentiformis, 3. to the cerebral peduncle by fibers which leave the lower aspect of the caudate and lentiform nuclei. The claustrum is a thin layer of gray substance situated on the lateral surface of the external capsule. Its transverse section is triangular, with the apex directed upward. Its medial surface, contiguous to the external capsule, is smooth, but its lateral surface presents ridges and furrows corresponding with the gyri and sulci of the insula, with which it is in close relationship. The claustrum is regarded as a detached portion of the gray substance of the insula, from which it is separated by a layer of white fibers, the capsula extrema, band of Ballarger. Its cells are small and spindle-shaped and contain yellowish pigment. They are similar to those of the deepest layer of the cortex. The nucleus amygdalae, amygdala, is an ovoid gray mass situated at the lower end of the roof of the inferior cornua. It is merely a localized thickening of the gray cortex, continuous with that of the uncus. In front, it is continuous with the putamen, behind with the stria terminalis and tail of the caudate nucleus. The internal capsule, capsula interna, is a flattened band of white fibers between the lentiform nucleus on the lateral side and the caudate nucleus and thalamus on the medial side. In horizontal section, it is seen to be somewhat abruptly curved with its convexity inward. The prominence of the curve is called the genu and projects between the caudate nucleus and the thalamus. The portion in front of the genu is termed the frontal part and separates the lentiform from the caudate nucleus. The portion behind the genu is the occipital part and separates the lentiform nucleus from the thalamus. The front part of the internal capsule contains 1. Fibers running from the thalamus to the frontal lobe, 2. Fibers connected to lentiform and caudate nuclei, 3. Fibers connecting the cortex with the corpus striatum, and 4. Fibers passing from the frontal lobe through the medial fifth to the base of the cerebral peduncle to the nucleus pontus. The fibers in the region of the genu are named the geniculate fibers. They originate in the motor part of the cerebral cortex, and after passing downward through the base of the cerebral peduncle with the cerebrospinal fibers, undergo decussation and end in the motor nuclei of the cranial nerves of the opposite side. The anterior two-thirds of the occipital part of the internal capsule contain the cerebrospinal fibers, which arise in the motor area of the cerebral cortex and, passing downward through the middle 3/5 of the base of the cerebral peduncle, are continued into the pyramids of the medulla oblongata. The posterior third of the occipital part contains 1. Sensory fibers largely derived from the thalamus, though some may be continued upward from the medial lemniscus. 2. The fibers of the optic radiation, from the lower visual centers to the cortex of the occipital lobe. 3. Acoustic fibers, from the lateral lemniscus to the temporal lobe. And 4. Fibers which pass from the occipital and temporal lobes to the nuclei pontus. The fibers of the internal capsule radiate widely as they pass to and from the various parts of the cerebral cortex, forming the corona radiata, and intermingling with the fibers of the corpus callosum. The external capsule, capsula externa, is a lamina of white substance situated lateral to the lentiform nucleus, between it and the claustrum, and continuous with the internal capsule below and behind the lentiform nucleus. It probably contained fibers derived from the thalamus, the anterior commissure, and the subthalamic region. The substantia innominata of Meinert is a stratum consisting partly of gray and partly of white substance, which lies below the anterior part of the thalamus and lentiform nucleus. It consists of 3 layers, superior, middle, and inferior. The superior layer is named the ansa lentiformis, and its fibers, derived from the medullary lamina of the lentiform nucleus, pass medially to end in the thalamus and subthalamic region, while others are said to end in the tegmentum and red nucleus. The middle layer consists of nerve cells and nerve fibers. Fibers enter it from the parietal lobe through the external capsule, while others are said to connect it with the medial longitudinal fasciculus. The inferior layer forms the main part of the inferior stalk of the thalamus and connects this body with the temporal lobe and insula. The stria terminalis, Taenia semicircularis, is a narrow band of white substance situated in the depression between the caudate nucleus and the thalamus. Anteriorly, its fibers are partly continued into the column of the fornix. Some, however, pass over the anterior commissure to the gray substance between the caudate nucleus and the septum pellucidum, while others are said to enter the caudate nucleus. Posteriorly, it is continued into the roof of the inferior cornu of the lateral ventricle, at the extremity of which it enters the nucleus amygdalae. Superficial to it is a large vein, the terminal vein, vein of the corpus striatum, which receives numerous tributaries from the corpus striatum and thalamus. It runs forward to the interventricular foramen and there joins with the vein of the choroid plexus to form the corresponding internal cerebral vein. On the surface of the terminal vein is a narrow white band named the lamina affixa. The fornix is a longitudinal arch-shaped lamella of white substance situated below the corpus callosum and continuous with it behind, but separated from it in front by the septum pellucidum. It may be described as consisting of 2 symmetrical bands, one for either hemisphere. The 2 portions are not united to each other in front and behind, but their central parts are joined together in the middle line. The anterior parts are called the columns of the fornix, the intermediate united portions the body, and the posterior parts the crura. The body, corpus fornicus, of the fornix is triangular, narrow in front and broad behind. The medial part of its upper surface is connected to the septum pellucidum in front and to the corpus callosum behind. The lateral portion of this surface forms part of the floor of the lateral ventricle and is covered by the ventricular epithelium. Its lateral edge overlaps the choroid plexus and is continuous with the epithelium covering of the structure. The undersurface rests upon the telochoroidea of the third ventricle, which separates it from the epithelial roof of that cavity and from the medial portions of the upper surfaces of the thalami. Below, the lateral portions of the body of the fornix are joined by a thin triangular lamina named a psalterium, lyra. This lamina contains some transverse fibers which connect the two hippocampi across the middle line and constitutes a hippocampal commissure. Between the psalterium and the corpus callosum is a horizontal cleft, the so-called ventricle of the fornix. Ventricle of Verga is sometimes found. The columns, columna fornicus, anterior pillars, fornicolumns, of the fornix arch downward in front of the interventricular foramen and behind the anterior commissure, and each descends through the gray substance in the lateral wall of the third ventricle to the base of the brain, where it ends in the corpus mammillare. From the cells of the corpus mammillare, the thalamo-mammillary fasciculus, bundle of Vic d'Azur, takes origin and is prolonged into the anterior nucleus of the thalamus. The columns of the fornix and the thalamo-mammillary fasciculus together form a loop resembling the figure 8, but the continuity of the loop is broken in the corpus mammillare. The column of the fornix is drawn by the stria medullaris of the pineal body and by the superficial fibers of the stria terminalis, and is said to receive also fibers from the septum pellucidum. Zuckerkandl describes an olfactory fasciculus which becomes detached from the main portion of the columns of the fornix and passes downward in front of the anterior commissure to the base of the brain, where it divides into two bundles, one joining the medial stria of the olfactory tract, the other joins the subcallosal gyrus, and through it reaches the hippocampal gyrus. The crura, crus fornicus, posterior pillars, of the fornix are prolonged backward from the body. They are flattened bands, and at their commencement are intimately connected with the undersurface of the corpus callosum. Diverging from one another, each curves around the posterior ends of the thalamus and passes downward and forward into the inferior cornu of the lateral ventricle. Here it lies along the concavity of the hippocampus, on the surface of which some of its fibers are spread out to form the alveus, while the remainder are continued as a narrow white band, the fimbria hippocampi, which is prolonged into the uncus of the hippocampal gyrus. The inner edge of the fimbria overlaps the fascia dentata hippocampi, dentate gyrus, from which it is separated by the fimbrio-dentate fissure. From its lateral margin, which is thin and ragged, the ventricular epithelium is reflected over the choroid plexus as the lateral projects to the choroidal fissure. Interventricular foramen, foramen of Monro. Between the columns of the fornix and the anterior ends of the thalami, an ovar aperture is present on either side. This is the interventricular foramen, and through it, the lateral ventricles communicate with the third ventricle. Behind the epithelial lining of the foramen, the choroid plexus of the lateral ventricles are joined across the middle line. The anterior commissure, precommissure, is a bundle of white fibers connecting the two cerebral hemispheres across the middle line and placed in front of the columns of the fornix. On sagittal section, it is oval in shape, its long diameter being vertical and measuring about 5 millimeters. Its fibers can be traced lateralward and backward on either side beneath the corpus striatum into the substance of the temporal lobe. It serves in this way to connect the two temporal lobes, but it also contains decussating fibers from the olfactory tracts. The septum pellucidum. Septum lucidum is a thin, vertically placed partition consisting of two laminae separating in the greater part of their extent by a narrow chunk or interval, the cavity of the septum pellucidum. It is attached above to the undersurface of the corpus callosum, below to the anterior part of the fornix behind, and the reflected part of the corpus callosum in front. It is triangular in form, broad in front and narrow behind. Its inferior angle corresponds with the upper part of the anterior commissure. The lateral surface of each lamina is directed towards the body and anterior cornu of the lateral ventricle, and is covered by the ependema of that cavity. The cavity of the septum pellucidum, cavum septum pellucidii, pseudocele, fifth ventricle, is generally regarded as part of the longitudinal cerebral fissure, which has become shut off by the union of the hemispheres in the formation of the corpus callosum above and the fornix below. Each half of the septum therefore forms part of the medial wall of the hemisphere and consists of a medial layer of gray substance derived from that of the cortex and a lateral layer of white substance continuous with that of the cerebral hemisphere. This cavity is not developed from the cavity of the cerebral vesicles and never communicates with the ventricles of the brain. The choroid plexus of the lateral ventricle. Plexus choroideus ventriculus lateralis, paraplexus, is a highly vascular, fringe-like process of pia mater which projects into the ventricular cavity. The plexus, however, is everywhere covered by a layer of epithelium continuous with the epithelial lining of the ventricle. It extends from the interventricular foramen, where it is joined with the plexus of the opposite ventricle, to the end of the inferior cornu. The part in relation to the body of the ventricle forms the vascular fringed margin of a triangular process of pia mater, named the telechoroidea of the third ventricle, and projects from under cover the lateral edge of the fornix. It lies upon the upper surface of the thalamus, from which the epithelium is reflected over the plexus onto the edge of the fornix. This portion, in relation to the inferior cornu, lies in the concavity of the hippocampus and overlaps the fimbria hippocampi. From the lateral ends of the fimbria, the epithelium is reflected over the plexus onto the roof of the cornu. It consists of minute and highly vascular villous processes, each with an afferent and an efferent vessel. The arteries of the plexus are A. The anterior choroidal, a branch of the internal carotid which enters the plexus at the end of the inferior cornu, and B. The posterior choroidal, one or two small branches of the posterior cerebral which passes forward under the splenium. The veins of the choroid plexus unite to form a tortuous vein which courses from behind forward to the interventricular foramen and there joins with the terminal vein to form the corresponding intercerebral vein. When the choroid plexus is pulled away, the continuity between its epithelial covering and the epithelial lining of the ventricle is severed, and a cleft-like space is produced. This is named the choroidal fissure. Like the plexus, it extends from the interventricular foramen to the end of the inferior cornu. The upper part of the fissure, that is the part nearest the interventricular foramen, is situated between the lateral edge of the fornix and the upper surface of the thalamus. Farther back, at the beginning of the inferior cornu, is between the commencement of the fimbria hippocampi and the posterior end of the thalamus, while in the inferior cornu, it lies between the fimbria in the floor and the stria terminalis in the roof of the cornu. The telechoroidea of the third ventricle. Telechoroidea ventriculus tertii, velum interpositum. Is a double fold of pia mater, triangular in shape, which lies beneath the fornix. The lateral portions of its lower surface rest upon the thalami, while its medial portion is in contact with the epithelial roof of the third ventricle. Its apex is situated at the interventricular foramen. Its base corresponds with the splenium of the corpus callosum, and occupies the interval between that structure above and the corpora quadrigemini and pineal body below. This interval, together with the lower portions of the choroidal fissures, is sometimes spoken of as the transverse fissure of the brain. At its base, the two layers of the velum separate from each other and are continuous with the pia mater investing the brain in this region. Its lateral margins are modified to form the highly vascular choroid plexuses of the lateral ventricles. It is supplied by the anterior and posterior choroidal arteries already described. The veins of the telechoroidea are named the internal cerebral veins, venae galenae. They are 2 in number and run backward between its layers, each being formed at the interventricular foramen by the union of the terminal vein with the choroidal vein. The internal cerebral veins unite posteriorly in a single trunk, the great cerebral vein, vena magna galenae, which passes backward beneath the splenium and ends in the straight sinus. End of section 16. Of Gray's Anatomy Part 4. This is Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Forebrain Part 5. Structure of the cerebral hemispheres. The cerebral hemispheres are composed of gray and white substance. The former covers their surface and is termed the cortex. The latter occupies the interior of the hemispheres. The white substance consists of medullated fibers varying in size and arranged in bundles separated by neuroglia. They may be divided according to their course and connections into 3 distinct systems. 1. Projection fibers connect the hemispheres with the lower parts of the brain and with the medulla spinalis. 2. Transverse or commissural fibers unite the 2 hemispheres. 3. Association fibers connect different structures in the same hemisphere. These are, in many instances, collateral branches of the projection fibers, but others are the axons of independent cells. 1. The projection fibers consist of efferent and afferent fibers uniting the cortex with the lower part of the brain and with the medulla spinalis. The principal efferent strands are 1. The motor tract, occupying the genu and the anterior thirds of the occipital part of the internal capsule, and consisting of A. the geniculate fibers, which decussate and end in the motor nuclei of the cranial nerves of the opposite side, and B, the cerebrospinal fibers, which are prolonged through the pyramid of the medulla oblongata into the medulla spinalis. 2. The corticopontine fibers, ending in the nuclei pontis. The chief afferent fibers are 1. Those of the lemniscus, which are not interrupted in the thalamus. 2. Those of the superior cerebellar peduncle, which are not interrupted in the red nucleus and thalamus. 3. Numerous fibers arising within the thalamus and passing through its stalks to the different parts of the cortex. 4. Optic and acoustic fibers, the former passing to the occipital, the latter to the temporal lobe. 2. The transverse or commissural fibers connect the two hemispheres. They include A. The transverse fibers of the corpus callosum, B. The anterior commissure, C. The posterior commissure, and D. The lyra or hippocampal commissure. They have already been described. 3. The association fibers unite different parts of the same hemisphere and are of two kinds. Those connecting adjacent gyri, short association fibers. 2. Those passing between more distant parts, long association fibers. The short association fibers lie immediately beneath the gray substance of the cortex of the hemispheres and connect together adjacent gyri. The long association fibers include the following: A. The uncinate fasciculus, B. The cingulum, C. The superior longitudinal fasciculus, D. The inferior longitudinal fasciculus, E. The perpendicular fasciculus, F. The occipitofrontal fasciculus, and G. The fornix. A. The uncinate fasciculus passes across the bottom of the lateral fissure and unites the gyri of the frontal lobe with the anterior end of the temporal lobe. B. The cingulum is a band of white matter contained within the cingulate gyrus. Beginning in front at the anterior perforated substance, it passes forward and upward parallel with the rostrum, winds around the genu, runs backward above the corpus callosum, turns around the splenium, and ends in the hippocampal gyrus. C. The superior longitudinal fasciculus passes backwards from the front lobe above the lentiform nucleus and insula. Some of its fibers end in the occipital lobe and others curve downward and forward into the temporal lobe. D. The inferior longitudinal fasciculus connects the temporal and occipital lobes, running along the lateral walls of the inferior and posterior cornues of the lateral ventricle. E. The perpendicular fasciculus runs vertically through the front part of the occipital lobe and connects the inferior parietal lobule with the fusiform gyrus. F. The occipital frontal fasciculus passes backward from the frontal lobe along the lateral border of the caudate nucleus and on the mesial aspect of the corona radiata. Its fibers radiate in a fan-like manner and pass into the occipital and temporal lobes lateral to the posterior and inferior cornua. Desjardins regards the fibers of the tapetum as being derived from this fasciculus and not from the corpus callosum. G. The fornix connects the hippocampal gyrus with the corpus mammillare and, by means of the thalamo-mammillary fasciculus, with the thalamus. Through the fibers of the hippocampal commissure, it probably also unites the opposite hippocampal gyri. The gray substance of the hemisphere is divided into 1. That of the cerebral cortex, and 2. That of the caudate nucleus, the lentiform nucleus, the claustrum, and the nucleus amygdalae. Structure of the cerebral cortex. The cerebral cortex differs in thickness and structure in different parts of the hemisphere. It is thinner in the occipital region than in the anterior and posterior central gyri, and is also much thinner at the bottom of the sulci than on the top of the gyri. Again, the minute structure of the anterior central differs from that of the posterior central gyrus, and areas possessing a special type of cortex can be mapped out in the occipital lobe. On examining a section of the cortex with a lens, it is seen to consist of alternating white and gray layers thus disposed from the surface inward: 1. A thin layer of white substance, 2. A layer of gray substance, 3. A second white layer, outer band of Ballinger or band of Generi, 4. A second gray layer, 5. A third white layer, inner band of Ballinger, 6. A third gray layer, which rests on the medullary substance of the gyrus. The cortex is made up of nerve cells of varying size and shape, and of nerve fibers which are either medullated or naked axis cylinders embedded in a matrix of neuroglia. Nerve cells. According to Cahal, the nerve cells are arranged in 4 layers, named from the surface inward as follows: 1. the molecular layer. 2. The layer of small pyramidal cells. 3. The layer of large pyramidal cells. 4. The layer of polymorphous cells. The molecular layer. In this layer, the cells are polygonal, triangular, or fusiform in shape. Each polygonal cell gives off some 4 or 5 dendrites, while its axon may arise directly from the cell or from one of its dendrites. Each triangular cell gives off 2 or 3 dendrites, from one of which the axon arises. The fusiform cells are placed with the long axis parallel to the surface and are mostly bipolar, each pole being prolonged into a dendrite which runs horizontal for some distance and furnishes ascending branches. Their axons, 2 or 3 in number, arise from the dendrites and, like them, take a horizontal course, giving off numerous ascending collaterals. The distribution of the axons and dendrites of all 3 sets of cells is limited to the molecular layer. The layer of small and the layer of large pyramidal cells. The cells in these 2 layers may be studied together, since, with the exception of the differentiation in size and the more superficial position of the smaller cells, they resemble each other. The average length in the small cells is from 10 to 15 microns, that of the large cells from 20 to 30 microns. The body of each cell is pyramidal in shape, its base being directed to the deeper parts and its apex towards the surface. It contains granular pigment and stains deeply with ordinary reagents. The nucleus is of large size and round or oval in shape. The base of the cell gives off the axis cylinder, and this runs into the central white substance, giving off collaterals in its course, and is distributed as a projection, commissural, or association fiber. The apical and basal parts of the cell give off dendrites. The apical dendrite is directed towards the surface and ends in the molecular layer by dividing into numerous branches, all of which may be seen, when prepared by the silver or methylene blue method, to be studied with projection bristle-like processes. The largest pyramidal cells are found in the upper part of the anterior central gyrus and in the paracentral lobule. They are often arranged in groups or nests from 3 to 5 and are named the giant cells of Betz. In the former situation, they may exceed 50 microns in length and 40 microns in breadth, while in the paracentral lobule, they may attain a length of 65 microns. Layer of polymorphous cells. The cells of this layer, as their name implies, are very irregular in contour. They may be fusiform, oval, triangular, or star-shaped. Their dendrites are directed outward, but do not reach so far as the molecular layer. Their axons pass into the subjacent white matter. There are 2 other kinds of cells in the cerebral cortex. They are A. The cells of Golgi, the axons of which divide immediately after their origins into large number of branches, which are directed towards the surface of the cortex. B. The cells of Martinati, which are chiefly found in the polymorphous layer. Their dendrites are short and may have an ascending or descending course, while their axons pass out into the molecular layer and form an extensive horizontal arborization. Nerve fibers. These fill up a large part of the intervals between the cells and may be medullated or non-medullated, the latter comprising the axons of the smallest pyramidal cells and the cells of Golgi. In their direction, the fibers may be either tangential or radial. The tangential fibers run parallel to the surface of the hemispheres, intersecting the radial fibers at right angles. They constitute several striata, of which the following are the more important. 1. A striatum of white fibers covering the superficial aspect of the molecular layer, plexus of Exner. 2. The band of Bechtereau in the outer part of the layer of small pyramidal cells. 3. The band of Generi, or external band of Ballinger, running through the latter of large pyramidal cells. 4. The interior band of Ballinger, between the layers of the large pyramidal cells and the polymorphous layer. 5. The deep tangential fibers, and the lower part of the polymorphous layer. The tangential fibers consist of A. The collateral of the pyramidal and polymorphous cells and the cells of Martinati. B. The branching axons of Golgi's cells. C. The collaterals and terminal arborizations of the projection, commissural, and association fibers. The radial fibers. Some of these, that is, the axons of the pyramidal and polymorphous cells, descend into the central white matter, while others, the terminations of the projection, commissural, or association fibers, ascend to end in the cortex. The axons of the cells of Martinati are also ascending fibers. Special types of cerebral cortex. It has already been pointed out that the minute structure of the cortex differs in different regions of the hemisphere. And A.W. Campbell has endeavored to prove, as the result of an exhaustive examination of a series of humans and anthropoid brains, quote, that there exists a direct correlation between the physiologic function and histologic structure, end quote. The principal regions where the typical structures departed from will now be referred to. 1. In the calcarine fissure and the gyri bounding it, the internal line of Ballinger is absent, while the band of generi is of considerable thickness and forms a characteristic feature of this region of the cortex. If a section be examined microscopically, an additional layer of cells is seen to be interpolated between the molecular layer and the layer of small pyramidal cells. This extra layer consists of 2 or 3 strata of fusiform cells, the long axis of which are at right angles to the surface. Each cell gives off 2 dendrites, external and internal, from the latter of which the axon arises and passes into the white central substance. In the layer of small pyramidal cells, fusiform cells identical with the above are seen, as well as ovoid or star-like cells with ascending axons, cells of Martinati. This is the visual area of the cortex, and it has been shown by J.S. Bolton that in old standing cases of optic atrophy, the thickness of Generi's band is reduced by nearly 50%. A.W. Campbell says, quote, histologically, 2 distinct types of cortex can be made out in the occipital lobe. The first of these coats the walls and bounding convolutions of the calcarine fissure and is distinguished by the well-known line of Generi or Vic d'Azur. The second area forms an investing zone a centimeter or more broad around the first and is characterized by a remarkable wealth of fibers, as well as by a curious piriform cells of large size richly stocked with chromophobe elements, cells which seem to have escaped the observation of Ramani Cajal, Bolton, and others who have worked at this region. As to the functions of these 2 regions, there is abundant evidence, anatomical, embryological, and pathological, to show that the first, or calcarine area, is that to which visual sensations primarily pass, and we are gradually obtaining proof to the effect that the second investing area is constituted for the interpretation and further elaboration of these sensations. These areas therefore deserve the name visio-sensory and visio-psychic, end quote. 2. The anterior central gyrus is characterized by the presence of the giant cells of Betz and by, quote, a wealth of nerve fibers immeasurably superior to that of any other part, end quote, Campbell, and in these respects differs from the posterior central gyrus. These 2 gyri, together with the paracentral lobule, were long regarded as constituting the, quote, motor areas, end quote, of the hemispheres. But Sherrington and Grumbaum have shown that in the chimpanzee the motor area never extends onto the free face of the posterior central gyrus, but occupies the entire length of the anterior central gyrus, and in most cases, the greater part or whole of its width. It extends into the depth of the central sulcus, occupying the anterior wall, and in some places the floor, and in some extending even into the deeper part of the posterior wall of the sulcus. 3. In the hippocampus, the molecular layer is very thick and contains a large number of Golgi cells. It has been divided into 3 strata. A. S. convolution, or S. granulosum, containing many tangential fibers. B. S. lacunosum, presenting numerous vascular spaces. C. S. radiatum, exhibiting a rich plexus of fibrils. The 2 layers of pyramidal cells are condensed into one, and the cells are mostly of large size. The axons of the cells of the polymorphous layer may run in an ascending, a descending, or a horizontal direction. Between the polymorphous layer and the ventricular ependema is the white substance of the alveus. 4. In the fascia dentata hippocampi, or dentate gyrus, the molecular layer contains some pyramidal cells, while the layer of pyramidal cells is almost entirely represented by small ovoid cells. 5. The olfactory bulb. In many of the lower animals, this contains a cavity which communicates through the olfactory tract with the lateral ventricle. In man, the original cavity is filled up by neuroglia, and its wall becomes thickened, but more and more so on the ventral than on the dorsal aspect. Its dorsal part contains a small amount of gray and white substance, but it is scanty and ill-defined. A section through the ventral part shows it to consist of the following layers from without inward. 1. A layer of olfactory nerve fibers, which are the non-medullated axons prolonged from the olfactory cells of the nasal cavity, and reach the bulb by passing through the cribriform plate of the ethmoid bone. At first they cover the bulb, and then penetrate it to end by forming synapses with the dendrites of the mitral cells, presently to be described. 2. Glomerular layer. This layer contains numerous spheroidal reticulated enlargements termed glomeruli, produced by the branching and arborizations of the processes of the olfactory nerve fibers with the descending dendrites of the mitral cells. 3. Molecular layer. This is formed of a matrix of neuroglia embedded in which are the mitral cells. These cells are pyramidal in shape, and the basal part of each gives off a thick dendrite which descends into the glomerular layer where it arborizes as indicated above, and others which interlace with similar dendrites of neighboring mitral cells. The axons pass through the next layer into the white matter of the bulb, and after becoming bent on themselves at right angle, are continued into the olfactory tract. 4. Nerve fiber layer. This lies next to the central core of neuroglia, and its fibers consist of the axons or afferent processes of the mitral cells passing into the brain. Some efferent fibers are, however, also present and end in the molecular layer, but nothing is known as to their exact origin. Weight of the encephalon. The average weight of the brain in the adult male is about 1,380 grams, that of the female about 1,250 grams. In the male, the maximum weight out of 278 cases was 1,840 grams, and the minimum weight 964 grams. The maximum weight of the adult female brain out of 191 cases was 1,585 grams, and the minimum weight 879 grams. The brain increases rapidly during the first 4 years of life and reaches its maximum weight by about the 20th year. As age advances, the brain decreases slowly in weight. In old age, the decrease takes place more rapidly, to the extent of about 28 grams per year. The human brain is heavier than that of any of the lower animals except the elephant and whale. The brain of the former weighs from 3.5 to 5.4 kilograms, and that of the whale, in a specimen 19 meters long, weighed rather more than 6.7 kilograms. Cerebral localization. Physiological and pathological research have now gone far to prove that a considerable part of the surface of the brain may be mapped out into a series of more or less definite areas, each of which is intimately connected with some well-defined function. Motor areas. The motor area occupies the anterior central and frontal gyri and the paracentral lobule. The centers for the lower limb are located on the uppermost part of the anterior central gyrus and its continuation onto the paracentral lobule. Those for the trunk are on the upper portion, and those for the upper limb on the middle portion of the anterior central gyrus. The facial centers are situated on the lower part of the anterior central gyrus, those for the tongue, larynx, muscles of mastication, and pharynx on the frontal operculum, while those for the head and neck occupy the posterior end of the middle frontal gyrus. Sensory areas. Tactile and temperature senses are located on the posterior central gyrus, while the sense of form and solidity is on the superior parietal lobule and precuneus. With regard to the special senses, the area for the sense of taste is probably related to the uncus and hippocampal gyrus. The auditory area occupies the middle third of the superior temporal gyrus and adjacent gyri in the lateral fissure. The visual area, the calcarine fissure and cuneus. The olfactory area, the Rhinencephalon. As special centers of much importance may be noted, the emissive center for speech on the left inferior frontal and anterior central gyri, Broca, the auditory receptive center on the transverse and superior temporal gyri, and the visual receptive center on the lingual gyrus and cuneus. End of section 17.
Of Gray's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Veronica Jenkins. Anatomy of the Human Body, Part 4, by Henry Gray. Composition and central connections of the spinal nerves. The typical spinal nerve consists of at least 4 types of fibers: the somatic sensory, sympathetic afferent or sensory, somatic motor, and sympathetic efferent or preganglionic. The somatic sensory fibers, afferent fibers, arise from cells in the spinal ganglia and are found in all the spinal nerves, except occasionally the first cervical, and conduct impulses of pain, touch, and temperature from the surface of the body through the posterior roots to the spinal cord, and impulses of muscle sense, tendon sense, and joint sense from the deeper structures. The sympathetic afferent fibers conduct sensory impulses from the viscera through the rami communicantes and posterior roots to the spinal cord. They are probably limited to the white rami connected with the spinal nerves in two groups, that is, the first thoracic to the second lumbar, and the second sacral to the fourth sacral nerves. The somatic motor fibers, afferent fibers, arise from cells in the anterior column of the spinal cord and pass out through the anterior roots to the voluntary muscles. The sympathetic afferent fibers probably arise from cells in the lateral column or the base of the anterior column and emerge through the anterior roots and white rami communicantes. These are preganglionic fibers which end in various sympathetic ganglia, from which postganglionic fibers conduct the motor impulses to the smooth muscles of the viscera and vessels, and secretory impulses to the glands. These fibers are also limited to two regions, the first thoracic to the second lumbar, and the second sacral to the fourth sacral nerves. The afferent fibers, which pass into the spinal cord, establish various types of connections, some within the cord itself for spinal reflexes, others for reflexes connected with higher centers in the brain, while still others conduct impulses of conscious sensation by a series of neurons to the cerebral cortex. The intrinsic spinal reflex paths. The collaterals and terminals of the ascending and descending branches of the posterior root fibers, which leave the fasciculus cuneatus to enter the gray matter of the spinal cord, end in various ways. Many end in the dorsal column, some near its apex, others in the substance of Rolando, others in the intermediate region between the dorsal and ventral columns. Others traverse the whole thickness of the gray matter to reach the ventral column, others end in the dorsal nucleus, and others pass through the gray commissure to the dorsal column of the opposite side. All of these collaterals and terminals end in connection with cells or dendrites of cells in the gray columns. The axons of these cells have various destinations. Some pass out into the lateral and ventral funiculi and turn upward to reach the brain. Those concerned with the intrinsic spinal reflexes come into relation either directly or indirectly with motor cells in the anterior column. It is very unlikely that either the terminals or collaterals of the dorsal root fibers affect simple direct connections with the motor cells of the ventral column. There is at least one, if not several, intercalated neurons in the path. These intercalated or correlation neurons may have short axons that do not pass out of the gray matter, or the axons may pass out into the proper fasciculi and extend for varying distances up and down or in both directions, giving off collaterals and finally terminating in the gray matter of the same or the opposite side. The shortest fibers of the proper fasciculi lie close to the gray matter. The longest ones are nearer the periphery of the proper fasciculi and are more or less intermingled with the long ascending and descending fasciculi, which occupy the more marginal regions of the spinal cord. Each sensory neuron with its ascending and descending branches, giving off as it does many collaterals into the gray matter, each one of which may form a synapse with one or several correlation neurons, is thus brought into relation with many correlation neurons, and each one of these in turn, with its ascending and descending branches and their numerous collaterals, is brought into relation either directly or through the intercalation of additional correlation neurons with great numbers of motor cells in the anterior column. The great complexity of these so-called simple reflex mechanisms in the least complex portion of the nervous system, the spinal cord, renders them extremely difficult of exact analysis. The association or correlation neurons are concerned not only with the reflex mechanism of the spinal cord, but play an equally important role in the transmission of impulses from the higher centers in the brain to the motor neurons of the spinal cord. The complex mechanisms just described are probably concerned not so much in the contraction of individual muscles as in the complicated action of groups of muscles concerned in the enormous number of movements which the limbs and trunk exhibit in the course of our daily life. Sensory pathways from the spinal cord to the brain. The posterior root fibers conducting the impulses of conscious muscle sense, tendon sense, and joint sense, those impulses which have to do with the coordination and adjustment of muscular movements, ascend in the fasciculus gracilis and fasciculus cuneatus to the nucleus gracilis and the nucleus cuneatus in the medulla oblongata. In the nucleus gracilis and nucleus cuneatus, synaptic relations are found with neurons whose cell bodies are located in these nuclei and whose axons pass by way of the internal arcuate fibers, cross in the raphe to the opposite side in the region between the olives, and turn abruptly upward to form the medial lemniscus or medial fillet. The medial fillet passes upward in the ventral part of the formatio reticularis through the medulla oblongata, pons, and midbrain to the principal sensory nucleus of the ventrolateral region of the thalamus. Here the terminals form synapses with neurons of the third order, whose axons pass through the internal capsule and corona radiata to the somatic sensory area of the cortex in the postcentral gyrus. Fibers conducting the impulses of unconscious muscle sense pass to the cerebellum partly by way of the fasciculus gracilis and fasciculus cuneatus to the nucleus gracilis and nucleus cuneatus. Thence neurons of the second order convey the impulses either via the dorsal external arcuate fibers directly into the inferior peduncle of the cerebellum, or via the ventral external arcuate fibers, which are continued from the internal arcuate fibers through the ventral part of the raphe, and, after crossing the midline, emerge on the surface of the medulla in the ventral sulcus between the pyramids, or in the groove between the pyramid and the olive. They pass over the lateral surface of the medulla and olive to reach the inferior peduncle, through which they pass to the cerebellum. Other fibers conducting impulses of unconscious muscle sense pass upward in the dorsal spinocerebellar fasciculus, which arises from cells in the nucleus dorsalis. The posterior root fibers conducting these impulses pass into the fasciculus cuneatus, and the collaterals from them to the nucleus dorsalis are said to come almost exclusively from the middle area of the fasciculus cuneatus. They form by their multiple divisions baskets about the individual cells of the nucleus dorsalis, each fiber coming in relation with the bodies and dendrites of several cells. The axons of the second order pass into the dorso-spinocerebellar fasciculus of the same side, and ascend along the lateral surface of the spinal cord and medulla oblongata until they arrive at the level of the olive. They then curve backward beneath the external arcuate fibers into the inferior peduncle and pass into the cerebellum. Here they give off collaterals to the dentate nucleus and finally terminate in the cortex of the dorsal and superior portion of the vermis, partly on the same side, but to a great extent by way of a large commissure to the opposite side. The fibers lose their myelin sheaths as they enter the gray substance, and terminate by end ramifications along the nerve cells and their processes. Some of the fibers are said to end in the nucleus dentatus and the roof nuclei of the cerebellum, the nucleus globosus, nucleus emboliformis, and nucleus fastigius, and others pass through them to terminate in the inferior vermis. A few fibers of the dorsal spinocerebellar fasciculus are said not to enter the inferior peduncle, but to pass with the ventral spinocerebellar fasciculus. The cerebellar reflex arc is supposed to be completed by the fibers of the superior peduncle, which pass from the cerebellum to the red nucleus of the midbrain, where some of their terminals and collaterals form synapses with neurons whose axons descend to the spinal cord in the rubrospinal fasciculus. The terminal and collaterals of this fasciculus end either directly or indirectly about the motor cells in the anterior column. The ventral spinocerebellar fasciculus, since most of its fibers pass to the cerebellum, is also supposed to be concerned in the conduction of unconscious muscle sense. The location of its cells of origin is uncertain. They are probably in or near the dorsal nucleus of the same and the opposite side. Various other locations are given: the dorsal column, the intermediate zone of the gray matter, and the central portion of the anterior column. The neurons of the first order, whose central fibers enter the fasciculus cuneatus from the dorsal roots, send collaterals and terminals to form synapses with these cells. The fibers which come from the opposite gray columns cross some in the white and some in the gray commissure, and pass with fibers from the same side through the lateral funiculus to the marginal region ventral to the dorsal spinocerebellar fasciculus. The fasciculus begins about the level of the third lumbar nerve and continues upward on the lateral surface of the spinal cord and medulla oblongata until it passes under cover of the external arcuate fibers. It passes just dorsal to the olive, and above this joins the lateral edge of the lateral lemniscus along which it runs, ventral to the roots of the trigeminal nerve, almost to the level of the superior colliculus. It then crosses over the superior peduncle, turns abruptly backward along its medial border, enters the cerebellum with it, and ends in the vermis of the same and the opposite side. Some of its fibers are said to join the dorsal spinocerebellar fasciculus in the medulla oblongata and enter the cerebellum through the inferior peduncle. A number of fibers are said to continue upward in the dorsolateral part of the tegmentum as far as the superior colliculus, and a few pass to the thalamus. They probably form part of the sensory or higher reflex path. The posterior root fibers conducting impulses of pain and temperature probably terminate in the posterior column or the intermediate region of the gray matter soon after they enter the spinal cord. The neurons of the second order are supposed to pass through the anterior commissure to the superficial anterolateral fasciculus tract of Gowers, and pass upward in that portion of it known as the lateral spinothalamic fasciculus. This fasciculus lies along the medial side of the ventro-spinocerebellar fasciculus. It is stated by some authors that the pain fibers pass upward in the anterolateral ground bundles. In some of the lower mammals, this pathway carries the pain fibers upward by a series of neurons, some of which cross to the opposite side, so that in part there is a double path. In man, however, the lateral spinothalamic fasciculus is probably the most important pathway. On reaching the medulla, these fibers continue upward through the formatio reticularis in the neighborhood of the median fillet to the thalamus, probably its ventrolateral region. Whether higher neurons convey the pain impulses to the cortex through the internal capsule is uncertain. The pathway is probably more complex, and Head is of the opinion that our sensations of pain are essentially thalamic. The pain and temperature pathways in the lateral spinothalamic fasciculus are not so closely intermingled but that one can be destroyed without injury to the other. Ransom suggests that the non-medullated fibers of the posterior roots, which turn into Lesor's tract and ascend or descend for short distances, not exceeding 1 or 2 segments, and finally end in the substantia gelatinosa, are in part at least pain fibers, and that the fasciculus of Lesor and the substantia gelatinosa represent part of the mechanism for reflexes associated with pain conduction and reception, while the fibers to the higher centers pass up in the spinothalamic tract. The fibers of tactile discrimination, according to Head and Thomson, pass up in the fasciculus cuneatus and fasciculus gracilis of the same side, and follow the path of the muscle sense fibers. The axons of the second order arising in the nucleus cuneatus and gracilis cross with the internal arcuate fibers and ascend to the thalamus with the medial lemniscus. Thence by neurons of higher order the impulses are carried to the somatic sensory area of the cortex through the internal capsule. The other touch fibers, shortly after entering the spinal cord, terminate in the dorsal column or intermediate gray matter. Neurons of the second order send their axons through the anterior commissure to pass upward in the antero-lateral funiculus, probably in the ventral spinothalamic fasciculus. In the medulla they join or pass upward in the neighborhood of the medial lemniscus to the thalamus, and thence by neurons of higher order to the somatic sensory area of the cortex. The remaining ascending fasciculi form a part of the complex known as the superficial anterolateral fasciculus tract of Gowers. The spinotectal fasciculus, as its name indicates, is supposed to have its origin in the gray matter of the cord and terminations in the superior and inferior colliculi of the midbrain, serving for reflexes between the cord and the visceral and auditory centers of the midbrain. The spino-olivary fasciculus (olivospinal, bulbospinal, Hellwig's bundle) is likewise of unknown constitution and function. There is uncertainty even in regard to the direction of its fibers. The sympathetic afferent fibers (visceral afferent, viscerosensory, splanchnic afferent) enter the spinal cord by the posterior roots of the thoracic and first 2 or 3 lumbar nerves, and the second to the 4th sacral nerves. The fibers pass to these nerves from the peripheral sympathetic system through the white rami communicantes. Some of the cell bodies of these afferent fibers are located in the spinal ganglia, and others are in the sympathetic ganglia. Some of the afferent sympathetic fibers end about the cell bodies of sensory neurons, and visceral impulses are thus transmitted to these neurons, which conduct them as well as their own special impulses to the spinal cord. Other sympathetic afferent neurons, whose cell bodies are located in the spinal ganglia, send collaterals to neighboring cells of somatic sensory neurons, and thus have a double path of transmission to the spinal cord. Such an arrangement provides a mechanism for some of the referred pains. These sympathetic afferent fibers presumably divide on entering the spinal cord into ascending and descending branches. Their distribution and termination within the spinal cord are unknown. Some of them probably eventually came into relation with the sympathetic afferent fibers whose cell bodies are located in the lateral column. Our knowledge concerning both the termination and origin of these fibers is very unsatisfactory. The sympathetic afferent fibers, splanchnic motor, visceromotor, preganglionic fibers, are supposed to arise from cells in the intermediate zone between the dorsal and ventral gray columns and in the intermediolateral column at the margin of the lateral column. These preganglionic sympathetic fibers are not distributed throughout the entire series of spinal nerves, but are confined to 2 groups, the thoracolumbar from the 1st thoracic to the 2nd or 3rd lumbar nerves, and the sacral group from the 2nd to the 4th sacral nerves. They pass out with the anterior root fibers and through the rami communicantes to end in sympathetic ganglia. The impulses are distributed from cells in these ganglia through postganglionic fibers to the smooth muscles and glands. The thoracolumbar outflow and the sacral outflow form 2 distinct functional groups, which are considered more fully under the sympathetic system. End of section 18. Recorded by Veronica Jenkins in Ottawa, Illinois.
Section 19 of Gray's Anatomy. Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Morgan Scorpion. Anatomy of the Human Body, Part 4 by Henry Gray. Composition and Connections of Cranial Nerves, Part 1. 4e. Composition and Central Connections of the Spinal Nerves. The cranial nerves are more varied in their composition than the spinal nerves. Some, for example, contain somatic motor fibers only. Others contain the various types of fibers found in the spinal nerves, namely, somatic motor, sympathetic efferent, somatic sensory, and sympathetic sensory. In addition, there are included the nerves of the special senses, namely, the nerves of smell, sight, hearing, equilibration, and taste. The hypoglossal nerve, 12th cranial, consists of somatic motor fibers only and supplies the muscles of the tongue. Its axons arise from cells in the hypoglossal nucleus and pass forward between the white reticular formation and the grey reticular formation to emerge from the anterolateral sulcus of the medulla. The hypoglossal nuclei of the two sides are connected by many commissural fibers and also by dendrites of motor cells which extend across the midline to the opposite nucleus. The hypoglossal nucleus receives either directly or indirectly numerous collaterals and terminals from the opposite pyramidal tract, corticobulbar or cerebrobulbar fibers, which convey voluntary motor impulses from the cerebral cortex. Many reflex collaterals enter the nucleus from the secondary sensory paths of the trigeminal and vagus, and probably also from the nervus intermedius and the glossopharyngeal. Collaterals from the posterior longitudinal bundle and the ventral longitudinal bundle are said to pass to the nucleus. The accessory nerve, 11th cranial, contains somatic motor fibers. The spinal part arises from lateral cell groups in the anterior column near its dorsolateral margin in the upper 5 or 6 segments of the cord. Its roots pass through the lateral funiculus to the lateral surface of the cord. It supplies the trapezius and sternocleidomastoideus. The cranial part arises from the nucleus ambiguus, the continuation in the medulla oblongata of the lateral cell groups of the anterior column of the spinal cord from which the spinal part has origin. The upper part of the nucleus ambiguus gives motor fibers to the vagus and glossopharyngeal nerves. The cranial part sends its fibers through the vagus to the laryngeal nerves to supply the muscles of the larynx. The root fibers of the cranial part of the accessory nerve pass anterior to the spinal tract of the trigeminal, while those of the vagus pass through or dorsal to the trigeminal root, and emerge in the line of the posterolateral sulcus. The nucleus of origin of the spinal part undoubtedly receives either directly or indirectly terminals and collaterals controlling voluntary movements from the pyramidal tracts. It is probable that terminals and collaterals reach the nucleus either directly or indirectly from the rubrospinal and vestibulospinal tracts. It is also connected indirectly with the spinal somatic sensory nerves by association fibers of the proper fasciculi. The cranial part receives indirectly or directly terminals and collaterals from the opposite pyramidal tract and form the terminal sensory nuclei of the cranial nerves. A few fibers of the cranial part are said to arise in the dorsal nucleus of the vagus, and are thus sympathetic efferent. They are said to join the vagus nerve. The vagus nerve, 10th cranial, contains somatic sensory, sympathetic afferent, somatic motor, sympathetic efferent, and taste fibers. The afferent fibers, somatic sensory, sympathetic, and taste, have their cells of origin in the jugular ganglion and in the nodosal ganglion, ganglion of the trunk, and entering the medulla divide into ascending and descending branches as do the sensory fibers of the posterior roots of the spinal nerves after they enter the spinal cord. 1. The somatic sensory fibers are few in number, convey impulses from a limited area of the skin on the back of the ear and posterior part of the external auditory meatus, and probably join the spinal tract of the trigeminal nerve to terminate in its nucleus. Connections are probably established to the central path of the trigeminal with the thalamus and somatic sensory area of the cortex for the conscious recognition of impulses. The descending fibers in the spinal tract of the trigeminal, terminating in the nucleus of the tract, probably establish relations through connecting neurons with motor nuclei in the anterior column of the spinal cord and with motor nuclei of the medulla. 2. The sympathetic afferent fibers are usually described as terminating in the dorsal nucleus of the vagus and glossopharyngeal. Some authors, however, believe they join the tractus solitarius and terminate in its nucleus. These afferent fibers convey impulses from the heart, the pancreas, and probably from the stomach, esophagus, and respiratory tract. Their terminals in the dorsal nucleus come into relation with neurons whose axons probably descend into the spinal cord, conveying impulses to the motor nuclei supplying fibers to the muscles of respiration, i.e. the phrenic nerve and the nerves to the intercostal and levatorius costarum muscles. Other axons probably convey vasomotor impulses to certain sympathetic efferent neurons throughout the spinal cord. The dorsal nucleus of the ala cinerea, and the posterior continuation of it into the commissural nucleus of the ala cinerea constitute probably the so-called respiratory and vasomotor center of the medulla. The shorter reflex neurons of the dorsal nucleus probably effect connections, either directly or indirectly, with motor cells of the vagus itself and other cranial nerves. 3. Taste fibers conducting impulses from the epiglottis and larynx are supposed to pass in the vagus and join the tractus solitarius, finally terminating in the nucleus of the tractus solitarius. It is not certain that this nucleus represents the primary terminal center for taste, and some authors maintain that the taste fibers terminate in the dorsal nucleus. The secondary ascending pathways from the primary gustatory nucleus to the cortex, as well as the location of the cortical center for taste, are unknown. A gustatory center has been described near the anterior end of the temporal lobe. The nucleus of the tractus solitarius is connected with motor centers of the pons, medulla, and spinal cord for the reactions of mastication and swallowing. 4. Somatic motor fibers to the cross-striated muscles of the pharynx and larynx arise in the nucleus ambiguus. This nucleus undoubtedly receives either directly or indirectly collaterals or terminals from the opposite pyramidal tract controlling the voluntary movements of the pharynx and larynx. The reflex pathways conveying impulses from the terminal sensory nuclei are unknown, but probably form part of the intricate maze of fibers constituting the reticular formation. 5. Sympathetic efferent fibers arise from cells in the dorsal nucleus of the ala cinerea. These are preganglionic fibers of the sympathetic system, and all terminate in sympathetic ganglia from which postganglionic fibers are distributed to various organs, i.e. motor fibers to the esophagus, stomach and small intestine, gallbladder, and to the lungs, inhibitory fibers to the heart, secretory fibers to the stomach and pancreas. The dorsal nucleus not only receives terminals of sympathetic afferent fibers for reflexes, but undoubtedly receives terminals and collaterals from many other sources, but the exact pathways are at present unknown. The glossopharyngeal nerve, 9th cranial, is similar to the vagus nerve as regards its central connections, and is usually described with it. It contains somatic sensory, sympathetic afferent, taste, somatic motor, and sympathetic efferent fibers. The afferent sensory fibers arise from cells in the superior ganglion and in the petrosal ganglion. The same uncertainty exists concerning the nuclei of termination and nuclei of origin of the various components as for the vagus. 1. The somatic sensory fibers are few in number. Some are distributed with the auricular branch of the vagus to the external ear, others probably pass to the pharynx and fauces. They are supposed to join the spinal tract of the trigeminal and terminate in its nucleus. The connections are similar to those of the somatic sensory fibers of the vagus. 2. Sympathetic afferent fibers from the pharynx and middle ear are supposed to terminate in the dorsal nucleus. Connections are probably established with motor nuclei concerned in chewing and swallowing. Very little is known, however, about the connections with other parts of the brain. 3. Taste fibers from the tongue probably terminate in the nucleus of the tractus solitarius. These fibers, together with similar fibers from the facial, nervus intermedius, and the vagus, are supposed to form the tractus solitarius and terminate in its nucleus. The central connections have been considered under the vagus. 4. Somatic motor fibers to the silopharyngeus muscle arise in the upper end of the nucleus ambiguus. The existence of these fibers in the roots of the glossopharyngeal is uncertain, as there are other paths by which such fibers might reach in the glossopharyngeal from the vagus. The sources of impulses passing to the nucleus ambiguus are considered under the vagus. 5. Sympathetic efferent fibres, motor and secretory fibres, arise from the nucleus dorsalis. Some authors believe that the secretory fibres to the parotid gland arise from a distinct nucleus, the inferior salivatory nucleus, situated near the dorsal nucleus. The preganglionic fibres from this nucleus terminate in the otic ganglion. The postganglionic fibres from the otic ganglion pass to the parotid gland. The acoustic nerve, 8th cranial, consists of two distinct nerves, the cochlear nerve, the nerve of hearing, and the vestibular nerve, the nerve of equilibration. End of Part 19. Section 20 of Gray's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Morgan Scorpion. Anatomy of the Human Body, Part 4 by Henry Gray. Composition and Connections of Cranial Nerves, Part 2. The cochlear nerve arises from bipolar cells in the spiral ganglion of the cochlea. The peripheral fibres end in the organ of Corti. The central fibres bifurcate as they enter the cochlear nucleus. The short ascending branches end in the ventral portion of the nucleus. The longer descending branches terminate in the dorsal portion of the nucleus. From the dorsal portion of the cochlear nucleus axons arise which pass across the dorsal aspect of the inferior peduncle and the floor of the fourth ventricle, the striae medullaris, to the median sulcus. Here they dip into the substance of the pons, cross the median plane, and join the lateral lemniscus. Some of the fibres terminate in the superior olivary nucleus. The fibres of the stria medullaris are not always visible on the floor of the rhomboid fossa. From the ventral portion of the cochlear nucleus, axons pass into the trapezoid body. Here some of them end in the superior olivary nucleus of the same size. Others cross the midline and end in the superior olivary nucleus of the opposite side, or pass by these nuclei, giving off collaterals to them, and join the lateral lemniscus. Other fibres either terminate in, or give off collaterals to the nucleus of the trapezoid body of the same or the opposite side. Other fibres from the ventral portion of the cochlear nucleus pass dorsal to the inferior peduncle and then dip into the substance of the pons to join the trapezoid body or the superior olivary nucleus of the same side. From the superior olivary nucleus of the same and opposite sides, axons join in the lateral lemniscus. Collaterals and probably terminals also pass from the lateral lemniscus to other nuclei in its path, and receive in turn axons from these nuclei. These are the accessory nucleus, the medial preolivary nucleus, the lateral preolivary or semilunar nucleus, and the nucleus of the lateral lemniscus. The trapezoid body consists of horizontal fibres in the ventral part of the formatio reticularis of the lower part of the pons, behind its deep transverse fibres and the pyramid bundles. The axons come from the dorsal and ventral portions of the cochlear nucleus. After crossing the raphe, where they decussate with those from the opposite side, they turn upward to form the lateral lemniscus. Fibres from the striae medullaris contribute to the trapezoid body. In addition, it sends terminals or collaterals to, and receive axons from, the superior olivary nucleus, the nucleus of the trapezoid body, the lateral preolivary or semilunar nucleus, and the mesial preolivary nucleus. The cochlear nucleus, the terminal nucleus for the nerve of hearing, is usually described as consisting of a larger dorsal nucleus on the dorsal and lateral aspect of the inferior peduncle, forming a prominent projection, the acoustic tubercle, and a ventral or accessory cochlear nucleus more ventral to the inferior peduncle. The two nuclei are continuous and are merely portions of one large nucleus. The axons from cells of the spiral ganglion of the cochlear nerve on reaching the nucleus divide into ascending and descending branches, which enter the ventral and dorsal nuclei respectively. Axons from the large fusiform cells of the dorsal nucleus pass partly by way of the stria medullaris to the trapezoid body and lateral lemniscus and the nuclei associated with the former, and partly transversely beneath the inferior peduncle and spinal tract of the trigeminal to the trapezoid body. Axons from the ventral cochlear nucleus pass partly by the stria medullaris, but for the most part horizontally to the trapezoid body. The superior olivary nucleus is a small mass of grey matter situated on the dorsal surface of the lateral part of the trapezoid body. Some of its axons pass backward to the adjacent nucleus. This bundle is known as the peduncle of the superior olivary nucleus. Other fibres from the nucleus join the posterior longitudinal bundle and terminate in the nuclei of the trochlear and ocular motor nerves. The majority of axons, after giving off collaterals to the nuclei itself, join the lateral lemniscus of the same side. Other axons pass in the trapezoid body toward the ventral portion of the cochlear nucleus. The nucleus of the trapezoid body lies between the root fibres of the adjacent nerve and the superior olivary nucleus. Its cells lie among the fibres of the trapezoid body. In it terminate fibres and collaterals of the trapezoid body, which come from the cochlear nucleus of the opposite and probably the same side, and from the opposite trapezoid nucleus. They terminate in the nucleus of the trapezoid body in diffuse arborizations and peculiar end plaques or acoustic calyces of yellowish colour which fuse with the cell bodies. Its cells are round and of medium size. Their axons pass into the trapezoid body, cross the median line, and probably join the lateral fillet. The lateral preolivary or semilunar nucleus lies ventral to the superior olivary nucleus. In it, end terminals and collaterals of the trapezoid body and probably fibres of the opposite cochlear nucleus. Its axons mingle with the trapezoid body and join the lateral fillet. The mesial preolivary nucleus is in contact with the ventral side of the nucleus of the trapezoid body. It receives many collaterals from the trapezoid body. Its cells are smaller than those of the trapezoid nucleus. Their axons join the lateral fillet. The lateral lemniscus, lateral fillet, the continuation upward of the central path of hearing, consists of fibres which come from the cochlear nuclei of the same and opposite side by way of the trapezoid body and from the preolivary nuclei. It lies in the ventral or ventrolateral part of the reticular formation of the pons, at first ventral, then lateral to the median fillet. Above the pons these ascending fibres come to the surface at the side of the reticular formation in the trigonum lemnisci and are covered by a layer of ependyma. This part of the lateral lemniscus is known as the fillet of Rayleigh. On reaching the level of the inferior colliculus, the dorsal fibres which overlie the superior peduncle decussate in the velum medullare anterius with similar fibres of the opposite side. Numerous small masses of cells are scattered along the path of the lateral lemniscus above the superior olivary nucleus, and constitute lower and upper nuclei of the lateral lemniscus. They are supplied with many collaterals and possibly terminals from the fibres of the lemniscus. The axons of the lower nucleus of the lateral lemniscus, which arise from the larger stellate or spindle-shaped cells, with long, smooth, much-branched dendrites, are said by some authors to join the lateral lemniscus. But according to Cahal, they pass medially toward the raphe. Their termination is unknown. The cells of the upper nucleus of the lateral lemniscus are more scattered. The same uncertainty exists in regard to their termination. The fibres of the lateral lemniscus end by terminals or collaterals in the inferior colliculus and the medial geniculate body. A few of the fibres are said to pass by the inferior colliculus to terminate in the middle portion of the stratum griseum of the superior colliculus, and are probably concerned with reflex movements of the eyes depending on acoustic stimuli. The inferior colliculi, lower or posterior quadrigeminal bodies, are important auditory reflex centres. Each consists of a compact nucleus of grey matter covered by a superficial white layer and separated from the central grey matter about by the aqueduct by a thin, deep white layer. Many of the axons which appear in the superficial white layer ascend through the inferior brachium to the medial geniculate body. Others, mainly from large cells in the dorso-mesial part of the nucleus, pass through the deep white layer into the tegmentum of the same and the opposite side and descend. Their termination is unknown, but they probably constitute an auditory reflex path to the lower motor centres, perhaps descending into the spinal cord with the tectospinal fasciculus. Other axons are said to descend in the lateral lemniscus to the various nuclei in the auditory path, held, and probably to motor nuclei of the medulla and spinal cord. The medial geniculate body receives terminals and collaterals from the lateral lemniscus, the central auditory path, and also large numbers of axons from the inferior colliculus of the same side, and a few from the opposite side. It is thus a station in the central auditory path. A large proportion of its axons pass forward beneath the optic tract to join the corona radiata, and then sweep backward and lateralward as the auditory radiation to terminate in the cortex of the superior temporal gyrus. V. Monaco holds that Golgi cells type II are interpolated between the terminations of the incoming fibres to the medial geniculate body, and the cells located there which give rise to the fibres of the auditory radiation. The medial geniculate bodies are united by the long, slender commissure of Gooden. These fibres join the optic tract as it passes over the edge of the medial geniculate and passes through the posterior part of the optic chiasma. It is probably a commissure connected with the auditory system. The vestibular nerve, vestibular root, 8th cranial, arise from the bipolar cells in the vestibular ganglion, Scarpa's ganglion. The peripheral fibres end in the semicircular canals, the saccule and the utricle, the end organs concerned with the mechanism for the maintenance of bodily equilibrium. The central fibres enter the medulla oblongata and pass between the inferior peduncle and the spinal tract of the trigeminal. They bifurcate into ascending and descending branches, as do the dorsal root fibres of all the spinal nerves and all afferent cranial nerves. The descending branches terminate in the dorsal, medial, vestibular nucleus, the principal nucleus of the vestibular nerve. This nucleus is prolonged downward into a descending portion in which end terminals and collaterals of the descending branch. The ascending branches pass to Dieter's nucleus, to Bechterer's nucleus, and through the inferior peduncle of the cerebellum to the nucleus tecti of the opposite side. The dorsal vestibular nucleus, medial or principal nucleus, is a large mass of small cells in the floor of the fourth ventricle under the area acoustica, located partly in the medulla and partly in the pons. The stria medullaris cross the upper part of it. It is separated from the median plane by the nucleus intercalatus. Its axons pass into the posterior longitudinal bundle of the same and the opposite side, and ascend to terminate in the nucleus abducens of the same side, and in the trochlear nucleus and the ocular motor nucleus of the opposite side, and to the motor nuclei of the trigeminal on both sides. The descending portion, the nucleus of the descending tract, extends downward as far as the upper end of the nucleus gracilis, and the decussation of the medial lemniscus. It is sometimes called the inferior vestibular nucleus. Many of its axons cross the midline, and probably ascend with the medial lemniscus to the ventrolateral region of the thalamus. The lateral vestibular nucleus, Dieter's nucleus, is the continuation upward and lateralward of the principal nucleus, and in it terminate many of the ascending branches of the vestibular nerve. It consists of very large multipolar cells whose axons form an important part of the posterior longitudinal bundle of the same and the opposite side. The axons bifurcate as they enter the posterior longitudinal bundle. The ascending branches send terminals and collaterals to the motor nuclei of the abducens, trochlear, and ocular motor nerves, and are concerned in coordinating the movements of the eyes with alterations in the position of the head. The descending branches pass down in the posterior longitudinal bundle into the anterior funiculus of the spinal cord as the vestibular spinal fasciculus, anterior marginal bundle, and are distributed to motor nuclei of the anterior column by terminals and collaterals. Other fibres are said to pass directly to the vestibular spinal fasciculus without passing into the posterior longitudinal bundle. The fibres which pass into the vestibular spinal fasciculus are intimately concerned with equilibratory reflexes. Other axons from Dieter's nucleus are supposed to cross and descend in the opposite medial lemniscus to the ventrolateral nuclei of the thalamus. Still other fibres pass into the cerebellum with the inferior peduncle, and are distributed to the cortex of the vermis and the roof nuclei of the cerebellum. According to Cahal, they merely pass through the nucleus fastigii on their way to the cortex of the vermis and the hemisphere. The superior vestibular nucleus, Bechterer's nucleus, is the dorsolateral part of the vestibular nucleus and receives collaterals and terminals from the ascending branches of the vestibular nerve. Its axons terminate in much the same manner as do those from the lateral nucleus. The facial nerve, 7th cranial, consists of somatic sensory, sympathetic afferent, taste, somatic motor, and sympathetic efferent fibers. The afferent or sensory fibers arise from cells in the geniculate ganglion. This portion of the nerve is often described as the nervus intermedius. 1. The somatic sensory fibers are few in number and convey sensory impulses from the middle ear region. Their existence has not been fully confirmed. Their central termination is likewise uncertain. It is possible that they join in the spinal tract of the trigeminal, as do the somatic sensory fibers of the vagus and glossopharyngeal. 2. The sympathetic afferent fibers are likewise few in number and of unknown termination. 3. Taste fibers convey impulses from the anterior 2/3 of the tongue via the chorda tympani. They are supposed to join the tractus solitarius and terminate in its nucleus. The central connections of this nucleus have already been considered. 4. Somatic motor fibers, supplying the muscles derived from the hyoid arch, arise from the large multipolar cells of the nucleus of the facial nerve. This nucleus is serially homologous with the nucleus ambiguus and lateral part of the anterior column of the spinal cord. Voluntary impulses from the cerebral cortex are conveyed by terminals and collaterals of the pyramidal tract of the opposite side, indirectly, that is, with the interpolation of a connecting neuron to the facial nucleus. This nucleus undoubtedly receives many reflex fibers from the various sources, i.e. from the superior colliculus via the ventral longitudinal bundle, tectus spinal fasciculus, for optic reflexes, from the inferior colliculus via the auditory reflex path, and indirectly from the terminal sensory nuclei of the brainstem. Through the posterior longitudinal bundle it is intimately connected with other motor nuclei of the brainstem. 5. Sympathetic efferent fibers, preganglionic fibers, arise according to some authors from the small cells of the facial nucleus, or according to others from a special nucleus of cells scattered in the reticular formation, dorsomedial to the facial nucleus. This is sometimes called the superior salivatory nucleus. These preganglionic fibers are distributed partly via the chorda tympani and lingual nerves to the submaxillary ganglion, thence by postganglionic vasodilator fibers to the submaxillary and sublingual glands. Some of the preganglionic fibers pass to the sphenopalatine ganglion via the great superficial petrosal nerve. The abducens nerve, 6th cranial, contains somatic motor fibers only, which supply the lateral rectus muscle of the eye. The fibers arise from the nucleus of the abducens nerve and pass ventrally through the formatio reticularis of the pons to emerge in the transverse groove between the caudal edge of the pons and the pyramid. The nucleus is serially homologous with the nuclei of the trochlear and ocular motor above, and with the hypoglossal and medial part of the anterior column of the spinal cord below. It is situated close to the floor of the fourth ventricle, just above the level of the stria medullaris. Voluntary impulses from the cerebral cortex are conducted by the pyramidal tract fibers, corticopontine fibers. These fibers probably terminate in relation with association neurons which control the coordinated action of all the eye muscles. This association and coordination mechanism is interposed between the terminals and collaterals of the voluntary fibers and the neurons within the nuclei of origin of the motor fibers to the eye muscles. The fibers of the posterior longitudinal bundle are supposed to play an important role in the coordination of the movements of the eyeball. Whether it is concerned only with coordinations between the vestibular apparatus and the eye, or with more extensive coordinations, is unknown. Many fibers of the posterior longitudinal bundle have their origin in the terminal nuclei of the vestibular nerve, and from the posterior longitudinal bundle many collaterals and terminals are given off to the abducens nucleus as well as to the trochlear and oculomotor nuclei. The abducens nucleus probably receives collaterals and terminals from the ventral longitudinal bundle, tectus spinal fasciculus, fibers which have their origin in the superior colliculus, the primary visual center, and are concerned with visual reflexes. Others probably come from the reflex auditory center in the inferior colliculus and from other sensory nuclei of the brain stem. End of Part 20. Section 21 of Gray's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by Morgan Scorpion. Anatomy of the Human Body, Part 4, by Henry Gray. Composition and Connections of Cranial Nerves, Part 3. The trigeminal nerve, fifth cranial, contains somatic motor and somatic sensory fibers. The motor fibers arise in the motor nucleus of the trigeminal and pass ventrolaterally through the pons to supply the muscles of mastication. The sensory fibers arise from the unipolar cells of the semilunar ganglion. The peripheral branches of the T-shaped fibers are distributed to the face and anterior 2/3 of the head. The central fibers pass into the pons with the motor root and bifurcate into ascending and descending branches which terminate in the sensory nuclei of the trigeminal. The motor nucleus of the trigeminal is situated in the upper part of the pons beneath the lateral angle of the fourth ventricle. It is serially homologous with the facial nucleus and the nucleus ambiguus, motor nucleus of the vagus and glossopharyngeal, which belong to the motor nuclei of the lateral somatic group. The axons arise from large pigmented multipolar cells. The motor nucleus receives reflex collaterals and terminals, one from the trigeminal nucleus of the trigeminal of the same, and a few from the opposite side, via the central sensory tract, trigeminothalamic tract. 2. From the mesencephalic root of the trigeminal, 3. From the posterior longitudinal bundle, 4. And probably from fibers in the formatio reticularis. It also receives collaterals and terminals from the opposite pyramidal tract, corticopontine fibers, for voluntary movements. There is probably a connecting or association neuron interposed between these fibers and the motor nucleus. The terminal sensory nucleus consists of an enlarged upper end, the main sensory nucleus, and a long, more slender descending portion which passes down through the pons and medulla to become continuous with the dorsal part of the posterior column of the grey matter, especially the substantia gelatinosa of the spinal cord. This descending portion consists mainly of substantia gelatinosa, and is called the nucleus of the spinal tract of the trigeminal nerve. The main sensory nucleus lies lateral to the motor nucleus beneath the superior peduncle. It receives the short ascending branches of the sensory root. The descending branches, which form the tractus spinalis, pass down through the pons and the medulla on the lateral side of the nucleus of the tractus spinalis, in which they end by collaterals and terminals, into the spinal cord on the level of the second cervical segment. It decreases rapidly in size as it descends. At first it is located between the emergent part of the facial nerve and the vestibular nerve, then between the nucleus of the facial nerve and the inferior peduncle. Lower down in the upper part of the medulla, it lies beneath the inferior peduncle and is broken up into bundles by the olivocerebellar fibers and the roots of the 9th and 10th cranial nerves. Finally, it comes to the surface of the medulla under the tubercle of Rolando, and continues in this position lateral to the fasciculus cuneatus as far as the upper part of the cervical region, where it disappears. The cells of the sensory nucleus are of large and medium size, and send their axons into the formatio reticularis, where they form a distinct bundle, the central path of the trigeminothalamic tract, which passes upward through the formatio reticularis and tegmentum to the ventrolateral part of the thalamus. Most of the fibers cross to the trigeminothalamic tract of the opposite side. This tract lies dorsal to the medial fillet, approaches close to it in the tegmentum, and terminates in a distant part of the thalamus. From the thalamus, impulses are conveyed to the somatic sensory area of the cortex by axons of cells in the thalamus through the internal capsule and corona radiata. Many collaterals are given off in the medulla and pass from the trigeminothalamic tract to the motor nuclei, especially to the nucleus ambiguus, the facial nucleus, and the motor nucleus of the trigeminal. The somatic sensory fibers of the vagus, the glossopharyngeal, and the facial nerves probably end in the nucleus of the descending tract of the trigeminal, and their cortical impulses are probably carried up in the central sensory path of the trigeminal. The mesencephalic root, descending root of the trigeminal, arises from unipolar cells arranged in scattered groups in a column at the lateral edge of the central grey matter surrounding the upper end of the fourth ventricle and the cerebral aqueduct. They have usually been considered as motor fibers that join the motor root, but Johnston claims that they join the sensory root of the trigeminal, that they develop in the alar, not the basal lamina, and that the pear-shaped unipolar cells are sensory in type. The trochlear nerve, fourth cranial, contains somatic motor fibers only. It supplies the superior oblique muscle of the eye. Its nucleus of origin, trochlear nucleus, is a small, oval mass situated in the ventral part of the central grey matter of the cerebral aqueduct at the level of the upper part of the inferior colliculus. The axons from the nucleus pass downward in the tegmentum toward the pons, but turn abruptly downward before reaching it, and pass into the superior medullary velum, in which they cross horizontally, to decussate with the nerve of the opposite side, and emerges from the surface of the velum, immediately behind the inferior colliculus. The cells of the trochlear nucleus are large, irregular, and yellowish in colour. The nuclei of the two sides are separated by the raphe through which dendrites extend from one nucleus to the other. They receive many collaterals and terminals from the posterior longitudinal bundle, which lies on the ventral side of the nucleus. There are no branches from the fibers of the pyramidal tracts to these nuclei. The volitional pathway must be an indirect one, as is the case with the other motor nuclei. The oculomotor nerve, third cranial, contains somatic motor fibers to the obliquus inferior, rectus superior, levator palpebrae superioris, and rectus medialis muscles, and sympathetic efferent fibers, preganglionic fibers, to the ciliary ganglion. The postganglionic fibers connected with these supply the ciliary muscle and the sphincter of the iris. The axons arise from the nucleus of the oculomotor nerve, and pass in bundles through the posterior longitudinal bundle, the tegmentum, the red nucleus, and the medial margin of the substantia nigra in a series of curves, and finally emerge from the oculomotor sulcus on the medial side of the cerebral peduncle. The oculomotor nucleus lies in the grey substance of the floor of the cerebral aqueduct, subjacent to the superior colliculus, and extends in front of the aqueduct a short distance into the floor of the third ventricle. The inferior end is continuous with the trochlear nucleus. It is from 6 to 10 mm in length. It is intimately related to the posterior longitudinal bundle, which lies against its ventrolateral aspect, and many of its cells lie among the fibers of the posterior longitudinal bundle. The nucleus of the oculomotor nerve contains several distinct groups of cells which differ in size and appearance from each other, and are supposed to send their axons each to a separate muscle. Much uncertainty still exists as to which group supplies which muscle. There are 7 of these groups or nuclei on either side of the midline, and 1 medial nucleus. The cells of the anterior nuclei are smaller, and are supposed to give off the sympathetic efferent axons. The majority of fibers arise from the nucleus of the same side. Some, however, cross from the opposite side and are supposed to supply the rectus medialis muscle. Since oculomotor and abducens nuclei are intimately connected by the posterior longitudinal bundle, this decussation of fibers to the medial rectus may facilitate the conjugate movements of the eyes, in which the medial and lateral recti are especially involved. Many collaterals and terminals are given off to the oculomotor nucleus from the posterior longitudinal bundle, and thus connect it with the vestibular nucleus, the trochlear and abducens nuclei, and probably with other cranial nuclei. Fibers from the visual reflex center in the superior colliculus pass to the nucleus. It is also connected with the cortex of the occipital lobe of the cerebrum by fibers which pass through the optic radiation. The pathway for voluntary motor impulses is probably similar to that for the abducent nerve. The optic nerve, or nerve of sight, second cranial, consists chiefly of coarse fibers which arise from the ganglionic layer of the retina. They constitute the third neuron in the series composing the visual path, and are supposed to convey only visual impressions. A number of fine fibers also pass in the optic nerve from the retina to the primary centers, and are supposed to be concerned in the pupillary reflexes. There are in addition a few fibers which pass from the brain to the retina. They are supposed to control chemical changes in the retina and the movements of the pigment cells and cones. Each optic nerve has, according to Sulzer, about 500,000 fibers. In the optic chiasma, the nerves from the medial half of each retina cross to enter the opposite optic tract, while the nerves from the lateral half of each retina pass into the optic tract of the same side. The crossed fibres tend to occupy the medial side of each optic nerve, but in the chiasma and in the optic tract they are more intermingled. The optic tract is attached to the tuber cinereum and lamina terminalis, and also to the cerebral peduncle as it crosses obliquely over its under surface. These are not functional connections. A small band of fibres from the medial geniculate body joins the optic tract as the latter passes over it and crosses to the opposite tract and medial geniculate body in the posterior part of the chiasma. This is the commissure of Gooden, and is probably connected with the auditory system. Most of the fibres of the optic tract terminate in the lateral geniculate body, some pass through the superior brachium to the superior colliculus, and others either pass over or through the lateral geniculate body to the pulvinar of the thalamus. These end stations are often called the primary visual centres. The lateral geniculate body consists of medium-sized pigmented nerve cells arranged in several layers by the penetrating fibres of the optic tract. Their axons pass upward beneath the longer fibres of the optic tract, the taenia semicircularis, the caudate nucleus and the posterior horn of the lateral ventricle, where they join the optic radiation of Gratiole. They pass backward and medially to terminate in the visuosensory cortex in the immediate neighbourhood of the calcarine fissure of the occipital lobe. This centre is connected with the one in the opposite side by commissural fibres which course in the optic radiation and the splenium of the corpus callosum. Association fibres connect it with other regions of the cortex of the same side. The region of the pulvinar in which optic tract fibres terminate resembles in structure the lateral geniculate body. Its axons also have a similar course, though in a somewhat more dorsal plane. The superior colliculus receives fibres from the optic tract through the superior brachium. Some enter by the superficial white layer, stratum zonale, others appear to dip down into the grey cap, stratum cinereum, while others probably decussate across the midline to the opposite colliculus. Other fibres from the superior brachium pass into the stratum opticum, upper grey-white layer. Some of these turn upward into the grey cap, while others terminate among the cells of this layer. Since the superior colliculi appear to be the central organs concerned in the control of eye muscle movements and eye muscle reflexes, we should expect to find them receiving fibres from other sensory paths. Many fibres pass to the superior colliculus from the medial fillet, as the latter passes through the tegmentum, bringing the superior colliculus into relation with the sensory fibres of the spinal cord. Fibres from the central sensory path of the trigeminal probably pass with these. Part of the ventral spinocerebellar tract, Gowers, is said to pass up through the reticular formation of the pons and midbrain toward the superior colliculus and thalamus. The superior colliculus is intimately connected with the central auditory path, the lateral lemniscus, as part of its fibres pass the inferior colliculus and terminate in the superior colliculus. They are probably concerned with reflex movements of the eye depending on auditory stimuli. The superior colliculus is said to receive fibres from the stria medullaris thalamis of the opposite side, which pass through the commissura habenuli and turn back to the roof of the mid-brain, especially to the superior colliculus. By this path, both the primary and cortical olfactory centres are brought into relation with the eye muscle reflex apparatus. The fibres which pass to the nuclei of the eye muscles arise from large cells in the stratum opticum and stratum lemnisci, and pass around the ventral aspect of the central grey matter, where most of them cross the midline in the fountain decussation of Maynard, and then turn downward to form the ventral longitudinal bundle. This bundle runs down partly through the red nucleus in the formatio reticularis, ventral to the posterior longitudinal bundle of the midbrain, pons and medulla oblongata into the ventral funiculus of the spinal cord, where it is known as the tectospinal fasciculus. Some of the fibres are said to pass down with the rubrospinal tract in the lateral funiculus. Some fibres do not decussate, but pass down in the ventral longitudinal bundle of the same side on which they arise, unless possibly they come from the opposite colliculus over the aqueduct. From the ventral longitudinal bundle, collaterals are given off to the nuclei of the eye muscles, the ocular motor, the trochlea, and the abducens. Many collaterals pass to the red nucleus, and are probably concerned with the reflexes of the rubrospinal tract. The fibres of the tectospinal tract end by collaterals and terminals either directly or indirectly among the motor cells in the anterior column of the spinal cord. The superior colliculus receives fibres from the visual sensory area of the occipital cortex. They pass in the optic radiation. Probably no fibres pass from the superior colliculus to the visual sensory cortex. End of Part 21.